Today we'll be reading Unit 3, Lesson 2, Explorations 2 and 3, pages 166 to 172. Exploration 2, Evaluating Genetic Modification. Genetically engineered crops, especially those not intended for the human food supply, are common in the United States and other industrialized nations. For example, genetically modified soybean plants now make up more than 80% of all soybean crops around the world. Many of these soybeans are used for animal feed. The use of genetically modified crops is increasing. Some modified crops are hardier and more productive than non-modified crops. These cotton plants can change genes to resist the herbicides that farmers use to kill weeds. Why might farmers want to grow cotton plants that are resistant to herbicides? Because if the herbicide also kills the cotton plant, then you're not only going to kill the weeds, but you're also going to kill the cotton. So having cotton that's resistant to those herbicides will only kill the weeds and not the cotton. So your plant can still live and thrive because it's in a weed-free environment. Genetically modified organisms. Inserting foreign DNA into an organism results in a genetically modified organism, or GMO. These organisms are called transgenic because genetic modification allows genes to cross the normal barriers between species, trans meaning across. GMOs have applications in agriculture, scientific research, medicine, and other industries. For example, genetically modified soybeans provide oils for skincare products and protein for food products. Many people are cautious about GMOs. This has limited the planting of genetically modified crops in some countries. Some GMO crops allow for reduced use of pesticides. Others required increased pesticide use for a successful harvest. Adoption of crops genetically engineered for herbicide tolerance, United States, 1996 to 2016. The number of acres planted with genetically modified crops has increased since their introduction in the mid-1990s. So in our graph, we have the percentage of planted acres, and then we have three colored lines, purple, blue, and yellow. The purple is soybeans, the blue is cotton, the yellow is corn. From the years 1996 to 2016, you can see that each of the lines went up significantly by 2016. Question 11. According to the graph, which crop was the first to reach 75%? So you're going to go here, you're going to find your 75%, and it would be, here, 75%, it would be that purple line right there, so the soybeans. Question 12. There may be more support for the genetic modification of a crop if the crop is not used for human food. What pattern in the graph might be explained by this statement? A, the different adoption rates between cotton and soybeans. B, the different adoption rates between cotton and corn, or C, the current adoption level of soybeans, corn, and cotton. And the answer would be B. Cotton and soybeans are used mostly for other products, like cotton is used to make clothing typically, and soybeans are used in animal feed. So if it's not something that's directly linked to something we eat, which yes, we do eat animals, but the corn is something that if we modified, it's a genetically modified organism that we would eat directly. Like we're not getting it secondhand. Whereas the soybeans, we would be getting it secondhand through the animal. So if you look at the cotton especially, and look between the cotton, which is used to make clothing, and the corn, which is eaten right away, you can see that cotton, significantly the line went up much faster than the corn line did for genetically modified cotton, because people weren't as worried about it. So B would prove that genetic modification happens more in plants that aren't eaten by humans. Case study, glowing mosquitoes. Malaria and dengue fever are human diseases that are spread by mosquitoes. Scientists can insert a gene that causes mosquitoes to die if they do not receive a specific chemical. Scientists insert a fluorescent gene from jellyfish at the same time to track which mosquitoes have been successfully modified. Researchers release only modified male mosquitoes to the wild because male mosquitoes do not bite humans. When female mosquitoes in the wild mate with the modified males, the offspring inherit the modified gene and die without access to the chemical. This reduces mosquito populations and can slow the spread of some human diseases. However, the release of genetically modified mosquitoes into the wild raises concerns about the impacts on birds and bats that may eat the GMOs. So these mosquito larvae glow. Their cells have genes from jellyfish that make fluorescent proteins. So the impact of genetically modified mosquitoes. Effect on individuals. Human individuals experience reduced exposure to diseases carried by mosquitoes, including malaria and dengue fever. Effects on society. Society benefits from reduced healthcare costs associated with these two diseases. The money saved can be used for other beneficial projects in communities. Effects on the environment. The release of genetically modified mosquitoes might have negative impacts on birds or bats that feed on the mosquitoes. Case study, research mice. Some research mice are genetically modified for scientific research. Knockout mice have a gene in their DNA knocked out or disrupted. Scientists then observe the mice to see how their traits change. This helps scientists identify the gene's function. Humans and mice have similar DNA. So research with knockout mice can help scientists learn what human genes do. For example, knockout mice research has provided data about genes involved in cancer and anxiety in humans. However, knockout mice are not a perfect research tool. Knocking out an important gene can cause developmental problems in the mice. Also, data from knockout mice studies cannot always be applied to humans. So a gene has been knocked out in the mouse on the left. Scientists use knocked out mice to learn what genes do. So you have what is a normal color for the mouse, and this one is classified as a knockout mouse, so its fur color is lighter because they knocked out a gene. So question 13, identify whether the impacts described in the table relate to individuals, society, or the environment. So the first one, knockout mice stay in the lab to limit the chance of spreading modified genes to wild mice. This would impact the environment because you're containing them so they don't run into the wild. Next one, researchers use information from knockout mice to treat similar diseases in humans. This would be society, because humans as a whole would benefit from learning these genes to be able to treat diseases. And then the last one, knockout mice might be hurt by changes made through genetic material. This is individual because it deals with the mice, each individual mice itself. So not all the mice might have problems, but some of them might due to the change. Turn the page. Case study, pharmaceutical chickens. In 2015, officials in the United States approved the farming of genetically engineered chickens. The chickens produce a special protein in their eggs that can be used to treat a human disorder. The protein is isolated from the eggs and given to people who lack the protein in their bodies. Without this protein, humans cannot break down fat molecules. The condition is fatal to infants and causes heart disease in adults. There is no effective treatment for the disorder. Plants, bacteria, and other organisms have been genetically modified for medical purposes. Goats can be modified to produce a chemical in their milk that breaks down blood clots in humans. Using mammals to produce pharmaceutical chemicals in their milk can be effective. Milk production is a natural process, and the milking does not harm the animals. So chickens can be genetically modified to produce a pharmaceutical protein in their eggs. And then it shows you a picture of some chickens. So the impacts of genetically modified chickens. Effect on the individuals. Humans with a protein disorder will experience fewer deaths and reduced illness by receiving replacement proteins from the chicken eggs. Laying eggs is a natural process, so the chickens are not harmed. Effect on society. Some medical chemicals need cellular processes for production. Using GMOs to produce these chemicals provides medicines that cannot be made in a laboratory. Effect on the environment. The introduction of GMOs into communities can be risky. Scientists try to prevent the transfer of genetically modified DNA by isolating GMOs. Some people worry that modified genes will accidentally spread to wild species. Question 14. Which of the following are positive impacts that genetically modified chicken have on society? Circle all that apply. A. Production of pharmaceuticals that cannot be manufactured. This would be beneficial, so it's making medicine that we can't make artificially. B. Improved medical care for humans. This is also a benefit. C. Increasing the number of GMOs in the community. This can be either a negative or a positive, so that would not be an answer because not all GMOs are the same. Not all GMOs are created equally, and some of them can actually be really harmful. D. Production of proteins that cure all diseases. GMOs, gene therapy, inserting genes into another animal to change things is not going to be able to cure all diseases. There's never going to be a perfect cure for everything because diseases are so different from each other. So D is not an answer. So the answers would just be A and B. Question 15, discuss. The use of animals in scientific research can help society, but the rights of animals must be considered. How can you apply the concept of ethics to the subject of animal testing? So ethics applies to animal testing because animals are living things, and living things have rights. Some might also explain that like various cultures have different value systems when it comes to animals. Like in China, they'll sell dogs and cats for eating purposes, but here in the United States, dogs and cats are considered pets, so it would be illegal to kill them and use them as meat. So forms of research considered acceptable in one culture might be viewed as offensive in another. So testing on cats and dogs in China would probably be okay because they're considered as a food source, whereas testing on cats and dogs in the United States would probably be considered a no-no because they're considered as companions. So not all cultures or individuals value human life above animal life. So you're going to get those people that think that the animal should be sacrificed to save the human, and you're going to get those people that think that the animal should not be sacrificed for the human, that they have both have the right to live. Engineer it. We're skipping question 16. Evaluate impacts of genetically modified crops. Bacillus thuringiensis, BT for short, is a bacterium that makes proteins that kill pests. Scientists isolated the BT gene and inserted it into the DNA of corn seeds. The result is BT corn. BT corn is a GMO that is resistant to leaf-eating caterpillars, so corn can be genetically modified to protect the plants from caterpillar damage. So complete the table to explain how genetically modified corn might impact individuals, society, and the environment. So first up for individuals, it increases the amount of food that's available, but it could have negative effects on each individual's health. Society, it would improve profits for farmers. There would be more food for communities, but you might cause issues between people who are for GMOs and people who are against GMOs. You might cause some fighting. And then effect on the environment, it would reduce pollution that would be as a result from the pesticide use. But however, it might decrease the bird population who use those caterpillars to survive. So if you're having less caterpillars on your plants, you might end up having less birds in your environment too. So 18, would you grow genetically modified corn if you were a farmer? So that's just a personal opinion. It's like if you were a farmer, you might say yes, because it would depend on crops, because farmers depend on those crops for their livelihood. It's what provides them with money to survive, so to pay for things. So as a farmer, yeah, you might choose to grow genetically modified crops so you can grow more crops and less of them will get damaged and eaten, and so then you'll make more money. But you might say no as well because maybe you don't believe that genetically modified organisms should be used. Maybe you think it might end up doing more harm than good. It's honestly just question 18 is your personal opinion. Turn the page. Exploration 3, evaluating gene therapy. Genetic engineering is not limited to genetic modification using bacteria. Other microbes and molecules can also be used to insert or influence genes in similar ways. For example, viruses are used in genetic engineering. Viruses inject viral genes into a cell. The cell then produces viral proteins that are assembled into new viruses. Scientists can change viruses so that they inject desirable genes into cells instead of viral genes. So scientists often choose adenoviruses to deliver genes to target cells. These viruses inject their genetic material into a cell nucleus. Picture of it. So question 19. What might be some advantages and disadvantages of using viruses in genetic engineering to treat human diseases? Viruses would be advantageous because they can inject the material into the cells without damaging them, but it could end up causing like a big change later on. I mean, there's always going to be pros and cons to putting something foreign into something else. So like if you're using viruses, yes, they're not living, and the advantage is it could be awesome to, introduce that genetic material because then it can inject it into the human, but it could also cause, if the material is not injected correctly, it could cause problems in the human's health. There could be a lot of other things that could go wrong. Gene therapy. Advances in biotechnology allow scientists to treat certain diseases. Gene therapy is a technique that uses genes to treat or prevent diseases. Gene therapy may be used to insert helpful genes or to remove harmful genes. It may also be used to insert suicide genes that cause cell death. This is a promising treatment for cancer. Most gene therapies are still being tested and studied. To be successfully treated with gene therapy, a disease must have a genetic cause, and scientists must know which genes cause the disease. Also, the disease should involve one or only a few genes. Finally, the effective genes must be accessible for treatment for gene therapy to be successful. So the gene, it has to be a genetic disorder, so it has to be something you inherit. So it's caused by genes, by DNA. Then it has to be something it's only a few genes they have to change. So they don't want to have to rewrite the entire strand of DNA. So they only want it to be like a couple genes that they have to change. And then the other thing, they have to be able to get into those genes. Like it has to be easy to reach, not super difficult to find. So one gene therapy success story involves children born with immune cells, so this immunodeficiency disease, that do not work. A single gene normally active in bone marrow causes the disease. Therapy involves taking bone marrow cells from a patient, inserting the correct gene, and replacing diseased bone marrow cells with modified cells. Without treatment, children born with the disorder must live in germ-free plastic bubbles. Gene therapy allows these children to live healthy lives. So types of gene therapy. Gene therapy can involve adding a functional gene, blocking a malfunctioning gene, or causing a malfunctioning cell to die. So they show you an addition of a therapeutic gene to provide normal cell function. The addition of a therapeutic gene blocks a malfunctioning gene, providing normal cell function. And then addition of a harmful gene produces a lethal substance and the targeted cell dies. So they show you the adding, the blocking, and the causing it to die. So here's where they add something to cause health. Here's where they add something so it blocks something else so the cell is healthy. And here's where they add something so the cell will die. Methods of gene delivery. Gene therapy may involve direct or indirect delivery of therapeutic genes. In direct gene therapy, a therapeutic gene is inserted into a virus. Then the genetically modified virus is delivered directly to the target site. In indirect gene delivery, a therapeutic gene is inserted into a virus. Cells are extracted from the patient. The genetically modified virus is used to introduce the gene to the cells outside of the body. The genetically modified cells are delivered to the target site. So indirect gene delivery, 1, identify target cells. Cells receiving therapeutic genes must be in an isolated area. So basically they find the cells that they want to change and they take them out of the body. 2, prepare gene for delivery. A therapeutic gene is inserted into the virus. 3, remove cells for modification. So this is where they actually remove the cells from. So step 1 is when they identify the genes in the cells that they are targeting. 2 is when they actually make the genetically modified virus. 3 is when they take the cells out of the patient's body. 4, they would inject the virus into those cells to change them, and those cells would then be grown in a lab. 5, deliver the cells then to where it needs to go, the target site. So that's the picture shown there. So they identify the gene that they want, they make the virus, they put that virus, they take out cells and put them in a petri dish. They inject that virus into those cells. Once the cells are changed, they put those cells back into the virus, into the target site that they are aiming for. So 20. Why might scientists choose indirect delivery instead of direct delivery for gene therapy? A, indirect delivery method takes less time than direct delivery method. This is false. Direct would usually be faster because you're just making the virus and putting it into the body and letting the body do it by itself. But not always. It's not always faster. Direct is not always faster, but you don't know for sure. B, direct gene delivery is only possible if the target area is accessible. This is false. Direct gene delivery is possible even if the target area is not accessible. In fact, it's probably the preferred method if you can't get to the site that you need to put the gene into. C, genetically modified cells are easier to deliver to the target area than genetically modified viruses. We're not talking, it's really not going to be any easier. Adding new cells into an area or adding a virus. The virus itself, neither is really going to be any easier. And then D, indirect delivery allows scientists to make sure the cells have been correctly modified and are producing the protein. This is true because they can see it in the petri dish, they can observe it under a microscope and make sure that everything is going the way it's supposed to, whereas in the direct delivery method, you won't necessarily know the changes are happening until once they've already happened. So you won't know if it's being effective or not. 21. Why are diseases involving many different genes poor candidates for gene therapy? Well, if many genes are involved, genetic modification is then going to be more complicated, more time-consuming, and more expensive. So with so many genes involved, the likelihood of negative side effects happening with the treatment also increase because you're changing more than one thing. Turn the page. Impacts of gene therapy. Gene therapy that has the potential to treat many diseases. The field of gene therapy is still new. Advances in biotechnology are improving gene delivery methods. While the future of gene therapy looks promising, it faces many challenges. Gene therapy raises many ethical concerns about whether human DNA should be changed. Genetic engineering is also costly. Gene therapy may not be available to poorer populations. Gene therapy can disrupt the function of healthy genes in target cells, and long-term effects are uncertain. New genes often result from natural mutations. Over time, evolutionary forces will minimize genes and traits with negative effects. New genes resulting from genetic engineering do not go through the same evolutionary process. So cystic fibrosis is a genetic disease that causes persistent lung infections. Treatment includes inhaling medication through a nebulizer. Gene therapies are being tested for this disease. So question 22, what are the requirements a disease must meet for gene therapy to be an effective option for treatment? Circle all that apply. A, it must have a genetic cause. This is true. If you're using gene therapy, it has to be genetic. B, it should involve many genes. This is false. You don't want to be changing a lot of things because then you're more likely going to have problems later. C, target cells must be accessible. This is true. You must be able to get to where you need to put the new material. D, target cells must be found throughout the patient's body. No, because in there it's harder to access. It takes more time to put the new cells or the virus or that's been modified into the body because you have to inject it into multiple sites. So just A and C are the answers. So identifying a potential disease for gene therapy treatment, consider what scientists know about these three diseases. So the predisposition to have heart disease is inherited. The disease is influenced by other factors including age, diet, high blood pressure, and smoking. A single gene causes hemophilia. Hemophilia results in a lack of clotting proteins in the blood. Clotting proteins are produced in the liver. The lack of clotting proteins can lead to severe bleeding and joint damage. People with type 1 diabetes produce little or no insulin. Insulin is needed to move sugars from the blood into cells. Type 1 diabetes involves multiple genes. It is triggered by environmental factors. Which disease has the best potential for treatment with gene therapy? Defend your answer using evidence from the text and explain what criteria you used. So hemophilia would be the best because it has the least amount of genes and it's genetically inherited. So hemophilia is a genetic disorder caused by only one gene, and its effects are limited to a set of target cells, which are the liver cells, because that's where hemophilia starts and where it's produced. So the criteria used to make this choice would be the disease must have a genetic cause, it must involve only one or a few genes, and the cells that are involved must be accessible and by current technology. So diabetes wouldn't be good because it involves multiple genes and it's triggered by environmental factors. Heart disease wouldn't be good because it's not only genetic, but you can get heart disease if you're a certain age, or if you have a bad diet, or if you have high blood pressure, or if you were a smoker. So heart disease is not solely dependent. It's not caused entirely by genes. Your assignment today is just to continue working on your test once your test is done, you can just submit it. It is due on the 19th for you Black Day students and the 20th for you Wool Day students. And that will be your last assignment besides the fun activities I have planned for the end of the year.